So now we're going to get into the occurrence distribution methods. Essentially, until now, we're looking at range distribution. Now we're going to go into a bit of occurrence distribution. So I covered this a little bit earlier, but essentially the questions that you're asking that require these occurrence distribution methods are different than the ones that you ask when you're doing range distribution methods, right? So maybe you're interested in migration, or maybe what areas contain high priority resources, either migratory corridors or stopover sites. You could also potentially be interested in how likely an animal is to visit a location of interest. Could be like a crossing site. Maybe you're interested in anthropogenic impacts like wind farms or anything like that, and you want to see if it's likely to visit an area like that. And maybe you're interested in where an animal crossed a linear infrastructure or any sort of feature in the landscape, like a road or a railroad. Obviously, if you think about it, if you have a series of locations, let's say they're a minute apart, you can be relatively sure where the animal crossed because there's only so far the animal can travel. Obviously, you have to be aware of the movement capabilities of your species, but there's only so many options the animal has if the locations are very close in time. However, if they're very far apart in time, then you start having issues actually. Where did the animal cross this road, right? So there's more of an uncertainty area. Could have maybe got a bit north or maybe south when it crossed. We just don't have that information from the locations. And this is exactly what's being targeted by these occurrence distribution methods, right? Like I said, range, you're always looking at extrapolating space use into the future. You're not interested in these interpolations between data points, right? And that's where the occurrence distribution is. The main difference just for what we call these different areas that we're getting or the regions that we're getting. So typically for range distribution, we get the home ranges. If we're doing occurrence distributions, however, we're not really getting an area per se. That's not the point here. It's not really biologically meaningful. But we're getting these occurrence regions, right? The probability that animal pass through a certain point. So instead of looking at all realizations of a movement process that could occur, because again, that's what it's capturing, those future locations, we're interested in pinpointing the exact realization that did occur. So that's why if you plot them side by side, the method that estimates range distribution and the method that estimates occurrence distribution, they should look very different, right? The thing is, though, is that even though range distribution, like we've seen so far, especially with the autocorrelated kernel density estimators, they're sampling independent. This occurrence distribution here, however, it's not, right? So if we see this example here, where we start with a series of points that are 1 hour apart, and this is the occurrence distribution that we get out of it. So the points that are more likely for the animal to be will be lighter colors, right, going towards the yellow. So we get some actually already nice where the animal was potentially more likely to be moving between those points. But again, this is very close in time. We only have 1 hour difference between those locations. Obviously, if we start just as an example, subsampling just to see the impact here, now the occurrence distribution starts getting a little bit more blurry, right? We're less certain now about where the animal potentially crossed. And obviously, if we keep doing that, in this case, going from 2 to 4 hour sampling interval, obviously we're getting a bigger and bigger occurrence region, right? Where the animal likely was crossing. So as you can expect, if you keep doing this, eventually they will look very similar. And this is why if sometimes people are doing these occurrence distribution methods and they look at this and they're like, okay, this seems like a home range to me, so I'm going to use it. But again, the problem is the sampling interval that you are using for that. They just happen to, at times, give a similar result, but it doesn't mean they're actually doing the same thing. So this is the same thing just that I've been showing with the sampling frequencies, starting from, this is a very high resolution, 2 minutes, and this is low resolution and 13 hours difference between the points. So the range distribution, again, sampling independent, it kind of pretty much looks the same no matter what. The occurrence distribution will essentially collapse to the actual movement path. So if you think about it, the area that we're getting out of here at some point is basically going to be 0. So there's no meaning there to calculate the area. Because it's just going to, if you have perfect data, this is just going to collapse to the animals here and then went here. It's like you have perfect knowledge. So yeah, this is the same information, just essentially hundreds of simulations to show this impact. Of essentially, you have the sampling frequency. If it's very, very high, so lots of points in a short period of time, the accuracy goes way low for comparatively to the home range. If you're trying to use it to get the home range estimate, right? So essentially, at some point, it collapses to 0. And here, with the tracking duration, if we have higher and higher tracking durations, that's why it eventually kind of looks like the home range and gets you the same results, the more data you have. And this holds true because there was simulated information, but this is also true with actual data that's been collected. So each of these points is an actual like animal, so it's not simulated data. And we see the same exact pattern, right? Where they, at some point, they match, but obviously this is an artifact and not really meaning that the occurrence estimates are giving you an area that's biologically meaningful. So what's the method that is used within CTMM to estimate occurrence regions, right? So within CTMM, it's used this generalized time series screening framework. Maybe you have heard it before because it's typically used a lot for geostatistics and computer science. Maybe you've at least seen the word somewhere. But essentially, the idea here is that it interpolates between locations. And it has well-known statistical properties, so it's very easy to work with it. So as a typical example, you could have weather stations, and you collect the temperature at those weather stations, and then you can interpolate what the temperature would be like across an area based on those points. Informations. So this occurrence distribution and estimating occurrence regions, it encompasses both this kringing method within CTMM, but also burn and bridge movement models and the correlated run and walk from crawl library. So the good thing about kringing is that it not only provides you with this prediction surface, right, where the animal is likely to cross or occur or not, but also gives you a measure of confidence, right? And that's what's important here as well, because maybe we're not very sure. So this is just an example of gazelle locations, just showing in the y-axis the y displacement, and then with across time. So it's just a one-dimensional information here. But essentially, obviously, if the locations are very close, the confidence intervals are going to conform very tightly with the actual trajectory. But obviously, if they're far enough in time, there's more uncertainty there. So we won't be very sure. Obviously, if you think about it, if we have a location that was collected at day 10 versus day 20, you have 10 days, the animal could have gone a lot of different places. So within CTMM, it's pretty easy. Again, it's doing a lot of the information that we've already been doing. We're going to use the information from these model fits that are describing the movement of the animal. Essentially, we have information about the position and the velocity of the correlation. And that's what's kind of being used to solve an animal's location at times. And it's, again, conditional upon the data and the fitted model. So it's using that to kind of interpolate. It's not just doing a point A to point B and seeing, OK, clearly, it went from there directly. Now, there is information within the movement parameters that's kind of informed. Maybe it tends to move a bit more tortuously and does not go in a straight line. So it's using that information to kind of interpolate. But like I was saying, the occurrence region is sampling dependent. So you have to be careful to not lose local sources of variation, right? So this is just a visual example, so it's easy to understand. If you have a series of values, right, this is essentially would be the true values that are being collected, right? Or the true trajectory of the individual. And these are the sampling points, right? The first location, second location. And there was a point here where maybe the animal went further than we would expect based on the other series of locations. But since we're only collecting at certain points, what happens with the Kriging method is that it might lose this big source of variation, right? It doesn't really account for it. Maybe it decreases a little bit, but it still doesn't fully account. And you don't see that variation when you're actually looking at the interpolated data. So in this case, if you're really trying to, if this is your goal, if your goal is one of those questions that I was talking about earlier, in this case, considering sampling frequency is more important because you want to be sure that you are getting those local sources of variation, right? So in this case, with a higher sampling frequency, we're able to correctly interpolate and get that. So it's just something to be aware of, because at times there's only so much the movement model will fill in the blanks for. So you might lose some of that variation. And some of the things we can do with this occurrence distribution method, we can fill in missing gaps if this is something you're considering, because again, it's our specific event that you really want to fill out to understand what was going on there. You can use this method to fill in those gaps. You can project beyond the sampling period. So if you have, actually, let me just explain this first. So this figure here, we have the first, the A. This is with Brownian bridge interpolation. This is not the Kriging interpolation, that's B. And then we have essentially this gray area, which is not really well seen in the figure here. But essentially, this half is gray and this half is gray. So essentially, there's a point where we're kind of assuming if the animal goes in there, we lose signal. It's kind of almost simulating a habitat signal loss related, right? So if we're using the Brownian bridge interpolation, which is the A, it kind of interpolates fully just, it goes from that point above to the other one. Yes, we lose all information when the animal goes into the dark zone, right? However, if we use Kriging interpolation, because again, remember, this is conditional upon the data and the movement model, it kind of understands something about the movement behavior of the animal. So that's in blue, in the gray area, is the interpolated movement path. So it is, I mean, it's not perfect, obviously, but it's still getting some information about what the animal could have been doing in that area that wasn't detected with the locations. And this is a similar thing, just the difference is just instead of being an area where the signal is lost, it's just periodically we have gaps in the data, right? Maybe it's not very clear, but the red points will be locations and the blue is the interpolation, right? And the orange points are essentially the true location that we're being missed. So we can see here with the Brownian bridge interpolation, kind of misses out on some of those essentially like explorations beyond that we're not getting detected because of the missing gaps. And with the Kriging interpolation, like you see here, it's getting this one while it's in the Brownian bridge is not. And some of them is also a little bit better, right? So it's never going to be perfect, and it's going to decrease with your sampling frequency being lower, but it's still a better way to fill in the gaps than the conventional methods. And something else you can do is basically you can just keep simulating stuff based on conditioned upon your movement model and just get an idea of potential areas that the animal would have been using based on the movement model, but it's not for some reason. So maybe there's a difference there that you want to explore. Like the animal, according to the movement behavior, could have gone there and it was close enough to the home range to potentially be using it. Why is it not using it? Maybe there's something to be focused on it and see what the patterns are that are making that happen. Does anyone have any questions? Yeah.
So this model for the occurrences is still just considering the information that we have. We don't have any context about environment or anything else for it.
No, it's
So
just
it's just based
locations.
on the movement.
Yes.
Okay.
It's based on the movement model, and there's no information on the habitat is there or whatever. No. Yeah. Okay. So now we can actually see how this works. There should be another script. I think it's just occurrence number 4, CTMM for occurrence, I believe. I'll check in a second. Good. Okay, so everyone has the right script. Okay. Okay, so you'll be very familiar with all of these steps at this point. But the good thing, though, is that for this particular method, we don't need to worry about range residency, right? Because we're not estimating home range, so you don't need to worry about the range residency assumption. Okay. 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 So these are the same steps. Just, again, having to do a CTMM select and all of that, that's the same. And to actually do the occurrence distribution, pretty simple, the occurrence function. Same logic as everything else within CTMM, data, and then you have the model fit. And in this case, you want the 95% utilization distribution, which is the default anyway. It shouldn't take too long. Is anyone having issues?
It's not finding the list. So should we run the model?
Not finding the object, you mean?
It's not finding the AKD list.
You don't need that, I think. I think I just had that extra from something else. Because when you do the load function here, it should already have the fit object. So you don't need to do the AKD list one. You can delete that line. Yeah. I think I probably just left it there accidentally. we go. Okay, it's saying it's having trouble with the plots, but let's try again. Okay, there we go. Up and disappeared. Oh, it's because I. Okay. It was there for 1 second. I don't know if anyone saw it, but it was there. Okay, there we go. Now it's back. Okay. So that's the occurrence distribution based on the location for this individual. So you can see, obviously this is just drawing the 95 constant contour. There's a probability distribution in the background. But yeah, you can see some potential areas where the movement likely occurred, right? And I'm afraid to plot it with locations. Maybe it'll take much time, but we'll see. Okay, it should eventually also plot the occurrence distribution in the background, just being a bit slow today. Yeah, but the difference here with the argument is that I'm removing the grids, and I'm removing that contour. The color of that contour is NA, so it doesn't show it. Yeah, it might not really show, but it's the same. It would be the same. It would just be the occurrence distribution in the background. I don't know. Is anyone having trouble plotting it, or is just my computer decided? Okay. So if you want to essentially fill in the gaps, if I remember correctly, this should have a sampling interval of 1 hour apart, but we can just double check. Yeah, so the sampling interval of this individual is 1 hour between locations. So obviously this is gonna essentially fill in the blanks with this simulate function where you're feeding it the data and the model object fit. And then we're saying instead of 1 hour, I want every location to be 5 minutes apart. So it's going to essentially kind of interpolate between the locations and give you a new simulated Scylla for us. So it might not show up when I plot it, but that's how you would do it if you want 5 minutes apart. Might be easier just because, again, I don't need to run the whole thing. I'm just going to subset it just so maybe it can plot faster. So just for the first 50 points. Okay. I'm just going to actually just plot the... Okay. So it's essentially all the 5 minutes apart points that are being simulated. So obviously, if you can think about it, it started from 1 hour to 5 minutes, so there's going to be a lot of these, even though it just tried to incorporate the movement behavior a little bit, but there's a certain point where they can only fill in the gaps so much, right? And then it's kind of meaningless. But if you're just trying to kind of like maybe fill in the gaps a little bit, that's how you would do it. Okay, so the occurrence distribution here, essentially, if we directly fit a Brownian motion model, it essentially will output the same that would have with a BBMM, right? Because it's the same, because the Brownian bridge movement model has that assumption anyway. But if you do it within CTMM and just fit a Brownian motion directly, it'll give you the same output. Obviously, it's not necessarily correct because the movement model may not fit the Brownian motion, but this is just an example so you can see how that would look like. So this is basically because, again, remember, the Brownian motion as infinite diffusion, and it just keeps diffusing, it's not bounded. That's what that means there. We're just creating a movement model that does that and essentially forcing the data to conform to that. Yeah, so obviously, in this case, the output of area for the effective sample set of area is 0 because, again, we don't have a home range. We're forcing it to not have that range residency behavior. So this is doing the same thing. We're plotting the occurrence, but with that Brownian motion fit. And does that just as long as that's running, but I can at least explain the next function. So this function predicts. What it's basically doing is that, like I said, we can predict beyond the sampling periods. So it's if the data is, what was it, 8 months or something? No, for 5 months for this individual, we can predict it for a year, and we'll basically use the movement model to have a simulation that's conditioned on that. And what the animal could be doing, you know, the extent beyond the sampling tracking that we had. And just in case it's not clear, what this is doing is just creating a sequence that goes from 1 to 1 year, and as a point every day, just a sequence. So it knows, like you have to tell it exactly like what is the sequence of timestamps that you want. Yep.
So it's one including the existing your data.
Yeah.
And then because it stops at 8 months, then completing with prediction of the remaining 4 months.
Yeah.
Okay.
Yeah. If it's if you're doing, you can do a simulation that's not conditioned upon the data, just a movement model, and that basically would mean it would not follow the locations. But if you're conditioned also on the data, then it would follow the location that you have, and it would just do whatever is filling in the gaps or going beyond based on it. Yeah. Okay, so it's not easy to tell because, again, the occurrence distribution is kind of hard to tell with this screen. Maybe it's easier on your own screens. You can kind of see the connections a little bit better. But yeah, that's basically the occurrence distribution with the assumption of Brownian motion. I would plot them side by side, but I feel like it will take a bit because my computer, for some reason, is being slow on here. But yeah, you can run it on and see the difference between the two. But yeah, the main difference is obviously one assumes Brownian motion, the other one will use whatever movement model is most appropriate to the data. Yeah, and this is just quickly showing them side by side, but we saw that already in the presentation. Just the impacts of coarsening the data and truncating the data, but it's the same type of plots I was showing before. Okay, does anyone have any questions? Okay, so it could be a bit jarring because it's not really about occurrence distribution anymore. We'll go back a bit to the same topic of range distribution that we were talking about. But one of my subjects of interest is essentially study design. So I'll go over a little bit of how you can optimize your animal tracking projects a little bit, and what some general recommendations and essentially what we need to consider. Obviously, for some of you, we already have collected data, so hopefully I don't say, look, this is how you did it wrong. Hopefully that's not the case. But I think it's useful in general to have some idea, even for understanding for the next project, or maybe there's still ways to kind of figure it out with the stuff you have already. So typically what happens a lot, especially when people come to me, it's like, hey, here's some data I have. And I ask them, okay, but what do you want to do with it? Is that they want to know everything about it, which, hey, I get it. I also want to know everything. I'm curious as well, but obviously that's not always going to be what you can actually get, right? Unfortunately, most of the time I can't tell them, yes, you can do everything. But obviously, if you think about it, for any successful project that you consider, there's really a series of steps that it should theory be done in a specific order if you're trying to get the most possible out of your data, right? So you start by defining your research questions, like what exactly is your spatial temporal scale of interest? And this part is very important because it kind of allows you to choose the sampling design that is most appropriate for those research questions and to identify those scales that you're interested in. And obviously afterwards you are going to collect some animal tracking data, then you're going to analyze that data, and then you're going to assess whatever conclusions you get out of it. Now, this sounds very good. In practice, you sometimes skip a few steps, or maybe you just can't, because it's like maybe you don't know exactly, like it's a new species that's not been studied well. You're not sure even what's the behaviors that you're going to want to capture. Again, there's a lot of uncertainty in our field. So even if we do want as much information a priori before we start the project, that's typically not going to happen. And there's obviously going to be a lot of limitations according to the sampling design, right? What device you can even put on your animal and what you're able to capture there. And there's obviously to consider the cost of the devices, the challenges that you can have during deployment, like you can lose animals, you can lose devices stop working, just the technological limitations that can occur even after, like you thought your devices were going to work really well, but there's some issues there. And obviously, if you go to a person that is just there to like analyze data, maybe they can, I really like this quote, like to consult the statistician, it's kind of skipping ahead, I don't know why. To consult the statistician after an experiment is finished is often merely to ask him to conduct a post-mortem examination. He can perhaps say what the experiment died of. Okay, but in general, you can at least get a general idea of, are you interested in fine scale processes? Are you interested in large scale processes? Or maybe both. Maybe you want to get best of both worlds here. And typically for fine scale processes, you're looking at movement metrics, maybe potentially something like speed or distance, where you're just trying to capture how far an animal travels, maybe the rates that the distances are covered, something like that. Maybe interested in behavior energetics, indicators of anthropogenic disturbance, anything like that. Obviously, for large-scale processes, one of them is the home range. Interested in capturing this, that I've already talked about a lot, this lifetime use of an animal's area. And that's also, again, for protected area delimitation, human-wildlife conflict, a lot of different questions that you can ask about this. And obviously, it would be really nice if you can always get birth to death. So this is an example of a white stork that was tracked enough that we get a really, really detailed picture. Again, it started in 94 until 2006. So you have so much information about this one individual. I mean, that sounds pretty nice, but again, it's not always the case in reality. So I also really like this picture. This is from Nathan in the 2022 paper that just talks about movement ecology as a big data field. But basically, this is we have the same underlying data, but one with low resolution, 30 minutes interval, and one with high resolution, 5-second interval between locations for the same individual. And obviously, you can see that for the high resolution, we're getting a lot more tortuosity of the movement versus the lower resolution. So if you think about it, if we're specifically looking at are these individuals interacting with each other, and you only have very low resolution, you might miss those interactions. And you might even say, oh, these two individuals kind of explore space in the same way. And you're missing out on all this information where they could have interaction spots. And one of them is definitely more bold in the way it explores its environment than the other one. And the same thing here. Again, low resolution data versus high resolution data. And we're trying to see if this particular species does have potential avoidance of areas that have high traffic of shipping lanes, for example. And obviously, low resolution data, it seems like it doesn't care. It goes right through that area. But we can see once we look into the high resolution data, that is actually full on avoiding those areas that could be problematic, right? So it's important to always consider this sampling frequency in relation to what behavior or what you want to be detecting, what process you're trying to detect. And obviously, this is all connected, especially for animal movement, with this idea of battery life of your devices if you have non-solar rechargeable devices, and also the fix rate, or if you're using a VHF tag, how many times you go out in the field to collect locations. This idea of sampling duration versus sampling frequency, and how that interacts with these spatial and temporal scales you're trying to identify. And obviously, if you have a GPS device, what happens as well is that you have kind of a choice between a series of parameters. Either you want maybe high duration, you want to track for as long as possible, but maybe that means for the same duration of the battery life that you need to have very low frequency, low resolution. But maybe you want high resolution, you want to detect those fine scales, but then maybe you can't track them for very long because you only get for very short durations, right? This is usually the curve that happens with a lot of models, GPS models that are currently available. The good thing, though, is that we have prior information, and we go through this understanding that we've been talking about all day, about animal movement being a realization of these continuous stochastic processes, and that we can summarize behavior with these characteristic timescales, but the position autocorrelation that we've been talking about a lot, the home range crossing time, and the velocity autocorrelation timescale, what this can do is basically they inform, because by imposing constraints of the sampling design, they inform what we should be doing. So we know what must be met to have sufficiently large sample sizes. Yeah, so this is the key point here. If we can identify the spatial temporal patterns, we can make sure that we select the best sampling design to detect those scales that we're interested in. So the good thing is that as long as it doesn't crash immediately when we tried using it, I developed this Shiny application that basically is analogous to power analysis. You can basically determine optimal sampling parameters based on a few different common estimates that people are interested in. Currently, I have home range and speed and distance travel as the ones available. Obviously, it could potentially be expanded, but these are the kind of ones that are available there. And the important thing here is that it will help you get as much of a certain set of conditions that you have to get large effective sample sizes. And obviously, depending on the question that you're interested in. There's always going to be cases where that's just simply not possible, but it's better to know beforehand than spending all the time in the field and then realizing you can't really do it. Okay, so I already covered this idea of effective sample size and absolute sample size, so I'm just going to skip right over it. But the main idea here is that if we're trying to detect the space use, the large case patterns like the home range, is that we really need to focus on the interaction between this position autocorrelation timescale and the sampling duration. That's what's important if our focus is home range. We want to make sure that we have enough of these home range crossing times to get a reliable home range estimate. And on the other side, for fine scale, for specifically speed and distance, we're interested in this interplay between the sampling frequency and the velocity autocorrelation timescale. Once again, we want the sampling frequency to be high enough that we still have a good information to estimate both speed and distance from a data set that we have. So if it's not physically possible, because we've seen for the animal movement to be uncorrelated, the main questions are, can we detect a signature of these correlations? And is this data sufficient for any specific research questions we have? So for example, here we have a simulated tracking data set. This is a new location every day. That's the sampling interval. And the underlying the velocity autocorrelation is around 10 minutes. That's how long it takes for the velocity autocorrelation to decay from the data. However, because it was minutes between locations, we're missing out. This blue is essentially the true trajectory. We're missing out on all of these extra movements that are happening, because again, the animal is not moving in a straight line. So we must carefully consider the frequency of data collection if our goal here is to get that information out for the true distance that the animal is traveling. And obviously, if the movement is more and more tortuous, this velocity autocorrelation is shorter. That means that the animal will be even more doing some more moves back and forth. So the SPI will increase with the shorter velocity autocorrelation. So in this other case, if we have this is a simulated set for 6 months, and we have a position autocorrelation that's 8 months. So it's actually larger than the current tracking duration that we have access to. So we know that if we kept sampling and the animal, we kept collecting the location from the animal, it might not necessarily stay in the same place, and it would go a little bit further. So it's important to consider that if we're trying to look specifically at home range and consider the sampling duration. Now, typically is there some questions? Yeah.
Sorry, just a quick question.
Yeah.
And I'm going to try and formulate this so that it makes sense. So it seems like, it's very important for us to be very careful about, the frequency at which we're getting fixes. Is there like a threshold where you kind of go from you can answer fine scale questions to, more broad scale questions? Like if you're maybe trying to infer behavior from movement, right? Like obviously more, closely more close in time fixed rates are going to be better, but is there like a threshold that, I just feel like sometimes the literature's kind of a little bit all over the place. Is there like something like maybe a good rule of thumb that you're like, okay, I'm sure many of us are kind of in a similar situation where, okay, we have a project we're starting, we want to maximize the amount of data that we can collect, but, we also want the battery life to, be sustained for the entire duration. Is there like does that make sense? Is there like a threshold that you would recommend?
I understand the question. I mean, obviously it's a little bit dependent on the exact question and behavior trying to capture. But if it is related to this, essentially this, how linearly is the animal movement, the movement happening, right, in the background? So it's not really a rule of thumb of like, we should collect every 2 minutes or 5 minutes. It's more in relation to this velocity autocorrelation parameter for a species that you're studying on. And maybe you don't have access to your exact, like this this species that I'm working on here, but maybe there's one from the same genus, or maybe something that you can use kind of as a proxy, right? And typically for velocity, for speed and distance, you wanted the sampling frequency to at least be 3 times lower than the velocity autocorrelation. So if your velocity correlation, let's say it's 10 minutes, you want this, like for example, half an hour, you want your sampling frequency to be every 10 minutes. So you're still getting at least 3 points essentially for that. I'll show a plot that shows like essentially the accuracy that you get of this relation between the velocity autocorrelation and the speed error that you can get. Might give you a better idea of that, but it's just kind of a rule of thumb. Yeah.
Thank you.
So for most species, and obviously this is going to be sometimes case by case. Unfortunately, I can't give you an answer that works 100% of the time. But for some species, they have consistent like position autocorrelation parameters across different individuals, right? Obviously, some of them is slightly higher than others, but still in general, it maintains within a mean, right? So it's stable and conservative enough that they can use these parameters to get a good idea. So if someone was going to do another study on African buffaloes, they can use this information to inform the next study, because we have a decent enough estimate of the position autocorrelation parameters. So the way the application works in the background, it's very similar to what we've been looking at. Again, it's doing a lot of the same processes. It's using the CTMM as the baseline for a lot of these methods. But the main idea is that you either select or upload a data set, because again, we need these parameters to come out from somewhere. I mean, you can basically guesstimate these parameters. Maybe you have some idea that the species you're working on maybe has a potential home range crossing time of a day or 2. So you can kind of use that if you have literally no access to any other data. But typically the way this works is that you need to give it something to extract those values first. But once you do that and you extract those measures, it basically simulates the device, and we have some information that we can kind of get idea of the battery life and the fixed rate of a certain model. Again, this is easy to get either from the manufacturers or sometimes even from the website, because they say 6 months of duration with this fixed rate. So you can use that information, kind of simulate the device within the app. And then it basically, you select whatever sampling duration, sampling interval you're interested in exploring. And then it does a lot of data simulation and model refitting in the background to kind of get our effective sample sizes. And from that, it gives you essentially an estimate of either home range estimation with the KDEs, or speed and distance with this continuous time speed and distance, which we didn't really cover. But again, if people are interested, we can talk more about it afterwards, to actually estimate the speed and distance with, again, the all underlying continuous time movement modeling. And then in the end, it gives you kind of a report of like, this is how it will look like for that particular combination of sampling duration and sampling interval, and what you can expect from that sort of data. But yeah, but this is just a general recommendations. Again, if the goal is speed and distance, you're looking at fine scale, you just adjust your sampling interval to ensure that is of sufficient resolution to detect this velocity autocorrelation. If your goal is home range area estimation, you adjust the sampling duration until you can detect these position autocorrelation parameters enough times to have a good effective sample size. Again, if both, sometimes you can do both, and other times you really can't. There's only, if you remember that curve with the GPS, sometimes no matter where you land on that curve of duration versus the fixed rate of the device, you can't really answer both questions at the same time, so you have to make a decision. Obviously, that's not always the case. Sometimes you can answer both, so there's also a way to do that. And the latest version of the app, so the way I currently had it is you basically had one individual and you got the information for it. This simulated individual is what I would expect. but now it actually incorporates some population level inferences. So you can actually see how many individuals may be sufficient to get, for example, a mean home range that would be reliable for what you're trying to do. Okay, so just another series of just general recommendations. If you're trying to focus on range distribution methods, if you see an analysis that's ignoring this position autocorrelation, it will typically underestimate home range areas. And if you're trying to look at this occurrence distribution, if it's ignoring velocity autocorrelation, you're typically overestimating occurrence areas. So you're losing one or the other. For home ranges, if you want a relative target bias of around 5%, especially if you don't want to go into the bootstrapping point, you want to ensure you have at least around 4.5, but better 20 or more for the effective sample size if you want a reliable estimate. But once again, remember, you definitely don't want to get into the bootstrapping if you can help it, because it's just very computationally intensive. But yeah, so this is kind of a repeat, so I'll just keep with what we had. But it's just basically showing the AKDE, how it changes with duration, frequency, and home range crossing time. But yeah, so in general, if you have, essentially you want to be on this side, right? This is the sampling duration, starting from very low to going to higher. Again, this is 4,000 days. This is a sampling duration that's usually not even easy to get. And versus the home range crossing time, right? So you have a species that maybe has a home range crossing time of around 16 days, and you want to ideally be in the durations that are higher here. So we have low relative errors, right? So that's where you want to be on this plot. Yeah, and for meta-analysis, again, I've already talked about this. You want to at least have 2 or 3 observed home range crossings and at least 2 or 3 representative individuals for this mean home range areas. And this is the same plot, but for the speed and distance, which is kind of how I got that around 3 times, right? The sampling frequency versus the directional persistence, the tau v. So essentially, you have to make sure that you are in this side of the plot, right? Your sampling interval is lower than your directional persistence, so you can get a decent estimate of speed and distance. But yeah, around 3 times the directional persistence. Yeah. So ideally, this is obviously, if you're lucky enough, you can try to do a pilot data study and try to see if you can get some information before. Or maybe you know someone that has done a study with a similar species and you can use that information. Again, you should always consider this, what spatial temporal scales you're trying to capture relative to the questions that you have. And keeping it simple is better. Even sampling rates is better than getting too clever of like, okay, I only want to sample when it's doing this and not other times, because then it just gets more complicated to analyze. Obviously, within reason. And you can check it first with simulated data. You can use the application that I've developed to do that and get at least some idea. And obviously, maybe what you can also do, instead of trying to do everything with one individual, maybe you can choose different individuals to answer different questions, right? Some of them you do fine scale, some of them you do large scale. And maybe that's the best way to kind of get the best of both worlds. If you must sample unevenly, just again, keep it simple. Usually no more than 2 different sampling rates. Again, this is just general recommendations. I know it's never necessarily this easy. And again, you can simulate with an even sampling first and see if it still works. At least using sampling rates at integer multiples might be also helpful, because then again, you can resample more easily. And again, check for any artifacts and be careful. But yes, be realistic, because unfortunately, no, we can't answer everything all the time. Okay, so hopefully we'll try the app and it's not going to crash on everyone, but hey, we'll see. But does anyone have any questions up until this point? No. Okay. Yep. It's, yeah, over here. Over here, forward.
I was just thinking, most of us might have data sets that are already, well, the data is already collected and established in AniMove Bank. But can you use this tool to potentially validate the quality of your data to be able to do such analyses?
Yeah, you can do that. That's definitely a use for this as well. Yeah. Okay, so there's a script, but basically what the script does is just load the app. So hopefully you already have it installed, because it should be when you install the AniMove package, it should already be there. But I guess we'll find out if there's any issues there. Okay, so in theory, this should be enough for it to start. Though we'll find out if that happens for everyone. Okay. Okay, so is everyone at least at this point? Okay, that's good. Step 1 complete. Okay, so the way this works, I try to make it as straightforward as possible. Though if anyone has any suggestions, I'm definitely all ears. So the first thing you do is kind of select your workflow, right? In this case, we're not gonna upload our own data, but just select. The difference here is that this has essentially all the species that are within CTMM are available within the app. Obviously, this is more for testing purposes than anything, but maybe you are working on a species that could be relevant for the ones that are available here. But in most cases, you'll probably want to upload data. As long as the data is with the MoveBank format, it should not have any trouble reading in. But obviously, if there's any problems, let me know, and I'll make sure it's fixed in the app. So in this case, we can set our research questions. For example, right now I can set both. Like I want to have both home range and maybe speed and distance estimates for my purposes, right? And in this case, this is essentially the analytical target. Maybe you're interested in, for this particular individual, do I get a reliable estimate of these parameters? That's what that one means. Or maybe you want a mean estimate of the population, right? So maybe let's select that one. And then this is essentially the difference here is that, okay, I already have in mind I can only purchase 10 devices, for example. So I already have a set number. So I want to see what I can do with those 10 devices. This other option is determining the minimum amount. Essentially will give you the minimum amount of individuals that you need for these for these research questions, right? So that's the difference there. So let's see, what's more interesting to try? Which one do you want to set? Okay, set number. Okay, let's try that one. And this to propagate uncertainty, I'll have to change it a little bit, but essentially it's trying to, from the individual level of the data sets that we have, the parameter uncertainty that we get out of the data, if we can put that also into the simulated values, which is not necessarily going to happen very easily. So typically what happens here is that we'll use whatever mean estimates of the parameters that we get. If we don't do that propagating uncertainty. Okay, so this should basically only show the tabs that you need, so you go over them one by one. And it basically should tell you, like, okay, now you can move on to the next one and stuff like that. Okay, so this is selecting the data. So let's pick our friendly African buffaloes. We've been with them this long, so why not finish with them as well? Okay, so you might have to give it a second to just load the data in. Okay, there we go. And basically at this point, we want to use all these individuals to inform our simulations. We can pick less than that, so maybe there's one that we don't want to use to inform the simulations within the app. But in this case, we can pick all of them. That's the only difference there. And this is showing it was also in the presentation, the position autocorrelation parameters for all the individuals and the velocity autocorrelation parameters for all individuals. Again, the scale here is between 20 to 40, 50 minutes, so it's still within a similar spatial temporal scale. Yeah. And obviously, it goes without saying at this point, especially because we are trying to look into home range estimation, that the data that you're feeding into the app, if you're trying to do home range estimates, it makes sense that you're feeding data that is also fitting the assumption of range residency, right? So if it's not doing that, it's not going to work very well. So the idea here is that you have to validate and then extract all these parameters. If you remember, essentially we're extracting all the autocorrelation timescales and all the other measures that are relevant from the data so we can do some informed simulations. Yeah?
How did you do to show the graphs behind? I'm not seeing it.
You mean when it was showing the plots over here?
Yes, and down also.
Oh, down also. Okay, so it's now running something, so I can't change anything. Oh, there we go. But basically, did you select a species and then an individual?
Yep.
Okay, and then you did validate?
No.
Okay.
Okay, validate.
Okay, validate, and then it should, we might need to also do extract for them to show up properly. Yeah. And give it a little bit of time. Is it running at least, or?
It ran. It shows question marks.
Okay.
And information.
All right, now let me just go quickly look at what's the output there.
This is what I get.
Okay, so you have to go down? No, sorry, timescales. Yeah. Oh, it's not showing there. Can you click the other one? Okay, so that's a problem. Okay, yeah. That's probably on me and not on you, don't worry. Okay, maybe the plots are not showing for some people. Can at least raise a hand if, okay, there's a few. Okay. I'm not sure what's causing that. In theory, they should show up, but that's a bug in the app. Yeah, I'm sorry. I will fix it, and then we'll have a new version. But yeah, I can at least follow along this, and then we can figure out what the problem is in detail. Okay, so now that we have the extracted parameters, it also already tells you the effective sample size of this data. Obviously, we don't have the information of the sampling design that we want to check. And that will be the next tab, the sampling design tab. So in this case, let's put down that we're evaluating with a GPS or satellite logger. Okay. So here, we can also add some limitations. Maybe there's some location error that we want to account for, potential for tag failure. Maybe there's a particular problem that you might have and want to consider, or the fixed success rate that you get. But I'm not going to put any of those yet. So that's just additional things that you can account for. So in theory, you should be able to see something like this. Though for some people, it's not working, which is this generated, essentially plot for the device. It's similar to what I was showing before with the duration and the sampling frequency for this device, and showing the different combinations of parameters, of sampling parameters that we can evaluate, right? Because again, remember the battery life, if you have very high frequency data, it's going to run out very fast, versus if you have low frequency being collected. So in this case, we can see maybe we want, and it's not very clear in the, because it's very like bright, but there is information on the roughly estimated effective sample sizes for area and for speed. And the speed is the green one, and the area is the blue one. Obviously, for speed, we're always going to have way higher effective sample sizes than for area. But essentially, we're trying to maximize both, right? Because we're trying to get both home range and speed and distance. So ideally, we want to be somewhere over here, right? Because if we're too far on this side, maybe we'll lose home range because we have very low effective sample size for area, and also for speed, it's definitely lower as well. But then here we have the same problem just on the other side, right? So we want somewhere over here. So let's say we want one fix every hour, right? Just let's test that and see how that would look like for the estimates that we get. So we have to select that sampling, and then you validate and you run. Yeah, basically, if you have the issue with the plots not showing, I'm going to use you as a guinea pig the next week to try to figure out what the problem is. Because it doesn't happen on my computer, so it's hard to figure out what the problem. Okay, so that gives you some estimate of the runtime. I think it's useful because sometimes, obviously, it can show like, okay, it's going to take an hour for it to run. Because again, this is all, again, power analysis simulation based, so it can take a decent amount of time to run. But it's also an estimate. It's not perfect, so even if it says less than a minute, sometimes it takes a little bit more than that. But it shouldn't be too bad. But yeah, a few minutes at least to run. Any other questions at this point? I don't know where the... Yes? Okay. Okay, that's fine. I mean, we can also use those.
Oh, I can't get it to run if I don't select from the plot. Like the manual selection is failing.
The manual selection is also failing?
Yeah.
Yeah, let's see. Yeah, that's the problem of stuff that's still in development. It always has some bugs to work through. Okay, there we go. Okay, so now we have essentially a simulated data set that we can use. And it will also see, you already see here, again, this is based on the model fit that we are outputting from the simulation. Our effective sample size for area is already not very good, right? And our effective sample size for speed is also not super high, but it's not as problematic as this. Obviously, at this point, we could try different ones and adjust. And again, remember, maybe I'm just setting a specific ideal GPS device, but you can change it depending on the ones that you have available. But let's just to go through the process and the workflow, what happens with these ones. Okay, so this will tell you more information about the home range estimate based on these parameters. It shouldn't take too long. There we go. Okay, so this is what the gray in the background is essentially the true home range. Again, this is simulation, so we have access to the expected value based on these parameters. And this is the home range that we're getting from our current set of sampling parameters. And we can also see the high and low confidence intervals. So we have a lot of uncertainty here from these sampling parameters, right? And again, the relative error is in an estimation of 51%. The thing here as well at this point, we could, okay, maybe if we tracked more individuals, what would happen? I'm not going to run with many here because I feel like they would just be stuck with taking a long time to run, but then you can set it a specific time. And we basically repeat this a few times, and we get an idea of, with these parameters, what we can expect for 3 individuals, right? And again, because the idea here is that we're essentially trying to get a reliable mean home range estimate. So maybe essentially we're asking the application, is 3 individuals enough to get a decent mean home range? Yeah, should I give it like lower sampling duration so it wouldn't take as long? Oh well, too late. I should add a function so you can stop it if you realize you don't want to keep running it. Well, I'll do that afterwards. It might be useful.
First of all, I think this app is really cool. So that's awesome that you created this. I'm just wondering, what are the limitations to the app? Mm-hmm. And when would you recommend using this application versus going through the whole coding process?
The limitations are very much related to what data you are using to estimate these parameters. Obviously, ideally, you'd have data for the same species and hopefully within the same study area to get a really good idea of the potential parameter landscape that we're dealing with, and then everything afterwards is pretty reliable. Mm-hmm. Obviously, the further you get from that ideal, the more this becomes a bit complicated, right? Because it is, like, if you think about it, it can give you a good starting point, but there's always so far it can go if you're very far. Like if you're, maybe you don't have data for your species, but there's one from maybe the same genus or something like that that you can kind of use to see. It will be less reliable, and maybe you can try going through the process just to get some idea, but you shouldn't like, okay, I need to use this or won't be able to do whatever, home range estimation or something like that. So I think that's the main limitation. How confident are you that the data you're using to inform these simulations is comparatively to what you actually expect in the field yourself? Yeah.
Sorry, as she said, this is brilliant, but I was just wondering, it's a little bit out of topic. Have you ever thought about including a step when you do the design to actually assist a new scientist, a new researcher coming towards telemetry on guiding on the ethical side of putting certain tags instead of others? Because the moment that we have to decide a battery, and how many times maybe we need to redeploy on the same animal. The big problem is that often we have then ethical clearance that, or even just wants to do things properly, and there is a lack of reference to which we can say, well, I use that path. We use, generally for large carnivores, we use 2-3% of the animal as a maximum threshold of the weight you can put on.
Yeah.
But still, there is not really something that it's commonly accepted. I don't know if...
I agree. I mean, yeah, because it's not always about like what's sampling frequency and what's sampling duration, because it's like there's a whole other factors coming into play here, right? I mean, yeah, they're not really helping here. The idea is to kind of like slowly build some sort of like database that can be more like more and more useful for people who look at different factors and information. I like that idea, but that's a lot of work.
No, sure. No, exactly.
Maybe if I get more time to work on this project, I can do something like that. No, but I fully agree. I mean, it's definitely something that you need to consider beyond just like, okay, maybe this app tells you should track them for a year, but maybe the only way you track them for a year is with a device that maybe is not ethically correct to place on the animal, right? So yeah, take this with a grain of salt. This is just trying to assist you a little bit, but there's always going to be other limitations beyond, yeah.
No, that was really my feeling. Is it's an amazing starting point, and it's just on that maybe we can. Then
Yeah, you can
develop
build
other-
up.
Yeah.
Yeah, no, I'm definitely interested in exploring more of that side as well, because it's definitely another concern that we have. I mean, that's the reason why in the sampling design I have other factors that you can kind of input to deal with some constraints. I don't know if I had weights of the device there at that point, but that's something that can potentially incorporate as well. Like these are my limitations in terms of weights for the device. Yeah. Okay, so this is just showing, in this case, I only simulated 3 individuals and the different home ranges that we get for them. But obviously, in general, our underestimation is around 50%, which is pretty high. Again, this is our effective sample set that we're working with. So we can already see maybe for this particular, because again, this is what you're getting, right? You're looking at a sampling duration of 30 days because you're trying to sample every hour, right? Because that's the settings that we gave it to try to simulate. So for the speed and distance, hopefully it doesn't take too long to run. Okay. I really should have put less values here. But yeah, but it will be the same logic with the home range tab, right? It will give you some information of comparatively to the expected value, because again, simulation, so we know the expected true value that we expect. What are the distances and speeds that we would get from this particular set of parameters?
No. This has been built with Shiny, yeah?
Mm-hmm. Yes, yes. Is anyone trying with different parameters, or is still, there's also the issue with the plots, right? Okay. Well, I'll figure it out, don't you worry. I'll make it functional again. Okay, if this starts taking too long, I might cancel. I wasn't really paying attention to what I was inputting. Oh well. This is a great time for questions, you guys. So. A while. Yeah. I mean, it's the first Shiny app I built, so it was a learning process as well. But I mean, at least the idea with this, because it's going beyond just people that have coding capabilities and trying to make it accessible to anyone that wants to make some decisions regarding animal tracking projects. Which is why we went for the Shiny app instead of just being functions within the application or the package. Yeah. Okay, I'm just going to cancel this because, again, I'm not sure how long it's going to take because my computer is also being slower than it should be. And I'm just going to start again and do like very low values just so it's quick. I don't want us to have to just wait for things to run for no reason if the point is just to show how this works. Okay. What I'll also do there is just show for one individual so it's just slightly faster. Okay, going through the same process, just very quickly. Give it some time. And this is, like I was saying, you can feed it your own data and then validate. It can be used for that as well. You upload your data and you can see how it looks like. It's the same exact process. Nothing really changes about it. There we go. Okay, so this is the output from the speed and distance showing essentially the true trajectory and then the simulated trajectory and the error that we get, which is not too bad relatively, especially compared to the home range estimate that we're getting. But we can see this is for distance, and then you can see for speeds. So we would expect to be, this is the true value, so it's very close to the true value. And the idea here is that if you go to report, you can basically build all the information that we get from this. Basically, will tell you essentially the ideal sampling duration should at least be around here, and this is what you get. For the sampling interval, for speed and distance estimation, and what you get. And in this case, it's telling you that your current tracking regime may be insufficient for both. So because you have high CIs for the relative errors that we're getting up here. And obviously, if you want better information, you need to do more individuals. And that's where, if you remember when I was looking at doing multiple simulations, you will just build up on this and you get more reliable results. But yeah, so it basically will give you some information here already, just the general information that we get. Sorry it took so long. I should have run it with less stuff in the beginning. I was just not thinking about that. But yeah, so if anyone has any questions, and then we can also try to debug that issue that some people are having with the plots, because I'm not sure what's causing that. But yeah, we can go through that. But if anyone has any specific questions, let me know. Yeah? Okay. So that ends my part, at least. Hopefully I left some time for Chris to cover the interaction parts. But yeah, that's it for me, guys. Hopefully it was useful.